Hello, and welcome to Biology Explained. Today, we are going to be looking at cell organelles and their functions. Now, imagine a city, not just any city, but one that runs 24/7, producing energy, transporting materials, managing waste, and even controlling information flow. Now, here's the twist that city exists in your body. It's called the cell. And within the cell are tiny structures known as organelles, each with specialized roles that keep life running. So, what are cell organelles? Cell organelles are specialized subunits within a cell, each designed to perform a specific function necessary for survival. Let's break that down. Specialized means each organelle has a unique structure tailored to its job. Subunits means they are parts within a larger system, the cell. Function means they each contribute to maintaining life processes. Think of organelles as a department in a company. Some generate energy, some manufacture products, some handle wastes, and some control operations. The cell is the smallest unit capable of carrying out all life processes. This idea comes from cell theory, which states that all living organisms are made of cells. The cell is the basic unit of life, and all cells arise from pre existing cells. This means that every organelle inside the cell contributes to keeping that cell alive, and every cell contributes to keeping the organism alive. One of the most important ideas in cell biology is division of labor. Instead of one part doing everything, different organelles handle different tasks. Why is this important? Because it increases efficiency, it allows simultaneous processes, and it reduces cellular stress. For example, the nucleus stores genetic information, the mitochondria produces energy, and the ribosomes build proteins. Each organelle focuses on its role, just like workers in a factory. In biology, structure and function are tightly linked. The shape and composition of an organelle determines what it can do. For instance, the mitochondria have a folded inner membrane to increase surface area for energy reproduction. Ribosomes are made of RNA and proteins, enable them to assemble amino acids into proteins. This principle appears everywhere in biology. Organelles do not work in isolation. They function as an integrated system. For example, the nucleus sends instruction, DNA to RNA. The ribosomes read those instructions to build proteins. The endoplasmic reticulum modifies those proteins, and the Golgi apparatus packages and ships them. This is like a production line. If one organelle fails, the whole system can be affected. Let's look at types of cells and organelles. There are two main types of cells: the prokaryotic cells, who have no membrane bound organelles, they are simpler in structure. For example, we have the bacteria. I will have the eukaryotic cells. They contain membrane bound organelles, they are more complex and found in plants, animals, and protists. This tutorial focuses mainly on eukaryotic cells because they show clear specialization. Let's expand on some terms you must fully understand. Organelle. They are specialized structure within the cell that performs a specific function. Cytoplasm is a gel-like substance where organelles are suspended and chemical reactions occur. Membrane-bound organelles. They are organelles surrounded by a lipid membrane, allowing compartmentalization. Compartmentalization, it is the separation of cellular processes into different areas, increasing efficiency and control. Now, let's relate this to the real world. Understanding organelles is not just theoretical, it explains how diseases develop, how drugs work, and how energy is produced in the body, and how genetic information is expressed. For example, if mitochondria fail, energy production drops and fatigue and diseases occur. So far, we've established that cells are the basic units of life. Organelles are specialized structures within cells, and each organelle has a unique function. They work together as a coordinated system, and their structure determines their function. This foundation is critical because next we'll begin exploring individual organelles in depth, starting with the control center of the cell. At the center of almost every eukaryotic cell lies a structure that controls everything, not by force, but by information. That structure is the nucleus. It is a membrane bound organelle that houses the cell's genetic material, the DNA. Let's break this down further. Membrane bound means it is enclosed by a nuclear envelope. Genetic material exemplifies that DNA contains instructions for all cellular activities, while being the control center exemplifies that it regulates growth, metabolism, and reproduction. To fully understand the nucleus, we must examine its internal components. One, the nuclear envelope, which is a double membrane that surrounds the nucleus. It contains nuclear pores, and these pores regulate movements of materials in and out. Think of it as a security gate controlling access to sensitive information. Number two, chromatin, which is DNA in a loosely packed form. When the cell is not dividing, DNA exists as chromatin. When the cell divides, chromatin condenses into chromosomes. This allows DNA to both be accessible and organized. Number three, nucleus. It is a dense region inside the nucleus. It is responsible for producing ribosomal RNA. It assembles ribosome subunits. Without the nucleus, protein production would stop. Let's look at the DNA, RNA, and protein. These are the most important processes in biology. They are the central dogma of molecular biology. Let's simplify it. The DNA stores information, RNA carries that information, and protein performs the function. In these steps, we'll understand it further. Step one is the transcription, which happens inside the nucleus. The DNA is used as a template. A copy is made in the form of messenger RNA. This is like copying instructions from a master blueprint. Step two is the translation. It happens outside the nucleus. The mRNA, which is the messenger RNA, travels to the ribosomes. The ribosomes read the codes, and amino acids are assembled into proteins. This is where the instructions are turned into actual products. Now, let's look at ribosomes. They are the protein factories. They are non membrane bound organelles responsible for protein synthesis. They are made of ribosomal RNA and proteins. There are two types of ribosomes. We have the free ribosomes and the bound ribosomes. The free ribosomes float freely in the cytoplasm and they produce proteins used inside the cell, while the bound ribosomes attached to the rough endoplasmic reticulum. They produce proteins for secretion, cell membranes, and other organelles. Let's connect everything. The nucleus provides the instructions, the messenger RNA carries the message, the ribosomes read the instructions, and proteins are assembled. This is a highly coordinated step by step process. Now that we understand how the cell controls and produces proteins, the next question is how are these proteins? processed, modified, and transported within the cell because production is just the beginning. Delivery is everything. Excellent. Now we move deeper into the cell's internal logistics and transport network, where raw proteins become functional, packaged, and delivered. So far, we've seen how the cell creates instructions and protein building. But here's something important: producing protein is not the final step. Proteins must be folded correctly, modified chemically, packaged properly, and transported to the right destination. And that's where the cell's internal transport system comes into play. We'll look at the endoplasmic reticulum. The endoplasmic reticulum, or ER, is a network of membranes that extends from the nucleus throughout the cytoplasm. Think of it as a transport and processing factory. There are two types: the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough is called rough because it is studded with ribosomes. Its functions: it synthesizes proteins, especially those for export or membranes. It begins protein folding and adds initial modification. The rough endoplasmic reticulum ensures proteins are properly formed before moving on. Smooth endoplasmic reticulum are called smooth because it lacks ribosomes. Its functions: lipid and steroid synthesis, detoxification of drugs and poisons, and calcium ion storage, especially in the muscle cells. The smooth endoplasmic reticulum is crucial for maintaining chemical balance. Let's look at protein folding and quality control. Proteins must fold into precise 3D shapes to function properly. Misfolded proteins can become toxic, and ER has quality control systems. If a protein is defective, it is either repaired or destroyed. This prevents cellular damage. Let's look at the Golgi apparatus. It is the post office of the cell. Once the protein leaves the endoplasmic reticulum, it travels to the Golgi apparatus. The Golgi is a stack of flattened membranes responsible for modifying proteins, sorting them, and packaging them into vesicles. The Golgi has two main phases: the cis phase, which is the receiving side that receives proteins from the endoplasmic reticulum, and the trans phase, which is the shipping side that sends proteins to their destination. Let's look at protein modification in the Golgi. Golgi can add carbohydrates, add phosphate groups, and modify protein structure. These changes determine where the protein will go. Let's look at the vesicles. The vesicles are the transport vehicles. Protein moves through the cell inside vesicles. They are small membrane-bound sacs. They transport materials between organelles. They carry substances to the cell membrane and store molecules temporarily. In summary, the endoplasmic reticulum synthesizes and processes proteins and lipids. The Golgi modifies, sorts, and packages proteins. The vesicle transports materials within the cells. Together, they form the endomembrane system. Proper protein handling is essential for cell survival. Now we've covered how proteins are made, how they are processed, and how they are transported. But one question that is very critical remains: where does the cell get the energy to power all these processes? Because without energy, nothing works. Now that brings us to mitochondria, chloroplasts, and energy transformation. Every movement you make, every thought you have, every heartbeat, all of it requires energy. But where does that energy come from? Inside your cells are tiny power generators working non-stop. These are the mitochondria. And in plant, there's an additional energy system, the chloroplasts. The mitochondria are the powerhouse of the cell. The mitochondrion is a double membrane bound organelle responsible for producing ATP, the adenosine triphosphate, which is the cell's energy currency. Understanding the structure of the mitochondrion is key to understanding its function. Its outer membrane is smooth and permeable. It allows substances to enter and exist. The inner membrane is highly folded into structures called the cristae. It increases surface area for energy production. Matrix is the inner fluid-filled space containing enzymes, mitochondrial DNA, and ribosomes. Mitochondria generate energy through cellular respiration. This is the process by which cells convert glucose and oxygen into adenosine triphosphate. There are three stages of cellular respiration: glycolysis in cytoplasm, Krebs cycle in mitochondrial matrix, and electron transport chain in the inner membrane. The final result is the ATP production. P is important because it is the currency of the cell. Powers muscle contraction, active transport, and protein synthesis. It also powers the nerve impulses. Without the ATP, the cell cannot function. Mitochondria have their own DNA, and this suggests they were once independent organisms. This idea is explained by the endosymbiotic theory, where ancient cells engulfed bacteria. Instead of digesting them, they formed a partnership, and over time, they became mitochondria, called chloroplasts. They are solar energy converters for plant cells only. In plant cells, energy can also come directly from sunlight, and this is the role of chloroplasts. Structure is a double membrane, internal stack called the thylakoids, and the thylakoids are arranged into grana with the surrounding fluid called the stroma. Chloroplasts perform photosynthesis, converting light energy into chemical energy through two main stages known as the light reaction and the Calvin cycle. The light reaction occurs in thylakoids. They capture strong light and they produce ATP and oxygen, while the Calvin cycle occurs in stroma and uses ATP to produce glucose. Here's the big picture. Plants produce glucose using sunlight, animals consume glucose, and mitochondria convert glucose into ATP. This creates a continuous energy cycle in nature. Case study is the cyanide poisoning. Cyanide blocks the electron transport chain in mitochondria. This stops ATP production completely. Even if oxygen is present, cells cannot use it, and this results in rapid energy failure and organ shutdown. In summary, mitochondria produces ATP through cellular respiration. ATP powers all cellular activities, and chloroplasts convert sunlight into glucose. Energy flows from sunlight to glucose to ATP. Disruption in energy production can lead to serious disease. We've now covered information control, protein production, transport systems, and energy generation. But there's still something missing. What about waste management, digestion, and structural support inside the cell? Because just like a city, a cell must clean itself, protect itself, and maintain its shape. That brings us to lysosomes, peroxisomes, vacuoles, and cytoskeleton. Imagine a city with no waste disposal system, no recycling, no structural framework, no storage facilities. It wouldn't last long. The same is true for the cell. Beyond production and energy, the cell must break down waste, detoxify harmful substances, store essential materials, maintain its shape and internal organization. Let's explore the organelles responsible for these critical functions. 1. Lysosomes. They are the digestive system of the cell. They are membrane-bound organelles filled with digestive enzymes. Let's look at the functions of lysosomes. They break down waste materials, digest worn-out organelles, and destroy harmful bacteria and viruses. This process is called intracellular digestion. They perform autophagy, which is self-eating. Cells can recycle their own components. Damaged organelles are enclosed in vesicles. Lysosomes break them down, and useful materials are reused. This maintains cellular health and efficiency. It is important to know that lysosomes contain powerful enzymes, and if they burst, they can digest the cell itself. This is sometimes referred to as programmed cell death. Peroxisomes, they detox and are the protection units. The peroxisomes are also membrane bound but have a different role. Functions of the peroxisomes include breaking down fatty acids, they detoxify harmful substances, and they produce and degrade hydrogen peroxide. They perform oxidative reactions. The peroxisomes use oxygen to carry out chemical reactions. They generate hydrogen peroxide and then convert it into water and oxygen. This prevents toxic buildup in the cell. Vacuoles, they are responsible for storage and regulations. Vacuoles are storage organelles. Their size and function vary between cells. In plant cells, they have large central vacuoles, which stores water, nutrients, waste products, while animal cells have smaller and are more temporary. They are used for storage and transport. Cytoskeleton, it is the structural framework. The cytoskeleton is a network of protein fibers that gives the cell its shape and organization. The cytoskeleton is made up of microfilaments with thin fibers involved in movement and shape. The intermediate filaments provide strength and stability. The microtubules help with transport, cell division, and movement. Example are the cilia and the flagella. They perform intracellular transport. The cytoskeleton acts like tracks for movement inside the cell. Vesicles travel along the microtubules. Motor proteins carry cargo. This ensures efficient delivery of material. In general summary, lysosomes breaks down wastes and recycle materials. Peroxisomes detoxify harmful substances. Vacuoles store materials, temperature, cytoskeleton provides structure and transport, and this maintain cellular health and stability. At this point, we've explored nearly every major organelle, but to truly master this topic, we must bring everything together. How do all these organelles interact as a complete system? How do they respond to real life biological challenges? How do all these organelles interact as a complete system? And how do they respond to real life biological challenges? We've explored each organelle individually, but here's the truth: no organelle works alone. A cell is not just a collection of parts, it is a coordinated dynamic system where every structure depends on the others. Now let's connect everything into one powerful unified picture: the cell as an integrated system. Think of the cell as a fully automated smart factory, where here's how everything works. The nucleus stores and sends instructions. The ribosome builds proteins based on those instructions. The endoplasmic reticulum processes those proteins. The Golgi apparatus modifies and packages them. The vesicles transport them. The cytoskeleton provides pathways for movement. The mitochondria supply energy. The lysosomes recycle the wastes, The peroxisomes detoxify harmful substances while the vacuoles store essential materials. Every process is linked. If one part fails, the entire system is affected. Working together, they all cause cellular homeostasis. Homeostasis means maintaining a stable internal environment. Cells must regulate temperature, pH level, energy levels, and waste accumulation. Organelles work together to maintain this balance. Let's walk through a complete example. The nucleus activates a gene, DNA is transcribed into messenger RNA. Messenger RNA travels to ribosomes. Ribosomes synthesizes a protein. Protein enters the rough endoplasmic reticulum. It is folded and modified and then sent to the Golgi apparatus, packaged into vesicles, transported via cytoskeleton, and released or used in cells. This is life happening at the cellular level. Let us see muscle contraction also for our example. When you move your arm, mitochondria produces ATP, cytoskeleton supports structure, calcium stored in the smooth endoplasmic reticulum is released, and protein interacts to cause contraction. Without ATP, no movement. Without the endoplasmic reticulum, no calcium regulation. With this, we can conclude that all life processes depend on the coordination of organelles from growth to repair to reproduction. Everything traces back to these microscopic structures. Some common student questions are: what is the most important organelle? Well, there's no single most important organelle. All organelles are interdependent. Can a cell survive if one organelle stops working? Sometimes temporarily, but long term failure leads to cell death. Why is this important for exams? Well, because it connects to genetics, physiology, diseases, biotechnology, and all of that. It's a foundational topic in biology. Let's summarize this in a powerful way. Cell is the basic unit of life. Organelles are specialized structures within the cell. Each organelle performs a unique function. The nucleus controls activities through DNA. Ribosomes synthesize proteins. The endoplasmic reticulum and Golgi process and transport materials. Mitochondria produces energy, which is the ATP. Chloroplasts convert sunlight into glucose. Lysosomes and peroxisomes handle waste and detoxification. Vacuoles store materials. The cytoskeleton maintains structure and movement. And all organelles work together to maintain life. You've made it this far. You now understand the cell not just as a structure, but as a living and functioning system, a microscopic world running with precision every second inside you. If you found this helpful, make sure to watch difficult sections, like and subscribe, and teach someone else. That's the best way to master it.